Welcome. In this video, we're going to discuss Chapter 7, and we're going to talk about the control of microbial growth. So just a couple things to keep in mind, thinking back historically, we talked about Ignaz Semmelweis and Joseph Lister before. If you remember talking about Semmelweis, we had made mention of the high risk of puerperal fever if a mother was to deliver in a hospital because of those physicians going back and forth between the morgue. We also made mention of Joseph Lister in the idea of surgical procedures and how he really helped to create this aseptic technique and utilizing these aseptic techniques during surgeries. So they were very instrumental in setting this stage for having this control of microbial growth. Now, before there were aseptic techniques, it was kind of noted that about 10% of all deaths were actually due to what is known as nosocomial infections. And nosocomial is a term that's sort of antiquated. Now we would call them HAI, healthcare acquired infections or healthcare associated infections. And that means that you went to the hospital and you actually picked up an infection in your stay. So about 10% of all deaths before aseptic technique were due to those infections. And especially, again, thinking back to that puerperal fever, delivering mothers were as high as 25% for that death rate. And again, that makes sense if you think about the idea of having an open wound and bleeding and all of those circumstances. It makes sense that that death rate was quite high. During the American Civil War as well, a surgeon may have cleaned their scalpel with a boot, or again, if we think about this idea of not using proper instrumentation or cleaning methods, it was very common that you would die of an infection. So this came up with the new idea of microbial growth. So we're going to discuss three specific terms, and you have to understand the differences and what organisms or what techniques you would use based upon the circumstance. So sterilization is the idea of being free of all life forms. And so you have nothing there. It is free of all life. And when we think about this, this means even resistant microbes. So this would be your viruses, this is your endospores, this is your fungi. It doesn't count for prions though, and if you remember discussing prions, they are misfolded proteins. Prions are highly resistant to all forms of sterilization, but the definition of sterilization implies that there's an absence of prions as well. When we think about disinfection, we think about the idea of removing pathogens. And usually, again, we think about the idea of having them be cytal. So there's microbial death, and this is usually happening to an inanimate object. So we think about our surfaces like cleaning our countertops or disinfecting our toilets, or maybe disinfecting some bottles that we're going to be drinking out of. The idea is to remove those pathogens. And so a lot of our cleaning products that we'll see claim to be a disinfectant. So they kill 99.9% of bacteria. And that would make sense. They get rid of those more susceptible or more moderate guys. But if you have something that's very resistant, more than likely it will take sterilization techniques. And then we have antisepsis. So disinfection is great for inanimate objects, but it's not good for human body surfaces. So if you think about a human body, we have to understand that those mechanisms like disinfection products aren't appropriate for us. So in order to remove pathogens from living tissue, we undergo what's known as antisepsis. These are what can safely be applied to body surfaces and are not going to cause damage. All right, so we're first going to make mention of physical methods, and then we'll talk about those chemical methods. So first that we're going to talk about is temperature. The most efficient sterilization technique is to utilize moist heat. And we would undergo what is known as autoclaving. And this is going to be steam under pressure. So it's 15 psi, which is pressure per square inch. You usually think about 121 degrees Celsius, and this can be anywhere from 20 to 45 minutes. But it kills all organisms and even those endospores. And if you remember, endospores are very hardy. They can live through some pretty extreme circumstances, but it will not kill prions. Prions are still relatively very resistant. And this is useful for a lot of things that can withstand those high heats and temperatures. So glassware, laboratory media, after we discard things in a lab, they should undergo this autoclaving technique. And anything that's going to be utilized intravenously should also be autoclaved. But usually when we think about heat, especially this moist heat is going to denature proteins. So remember, proteins have a very specific structure and a very specific function. If you apply extreme amounts of heat, it will misfold the protein or it'll completely unfold the protein. And that's what happens during this denaturation process. But heat can also preserve canned foods, and this is usually the most common method for food preservation. We can also have dry heat sterilization as well. And so when we think about dry heat, this would be like flaming or incineration. And this is very effective as well, but moist heat is the most efficient. So if you look down at the equivalent treatments in order to have a sterilization technique, with autoclaving, again, which is a moist heat, you need 121 degrees Celsius. And again, it can range depending on the time, but because of it being moist heat, it doesn't need as much time. Versus hot air sterilization, you need much higher temps in a long period of time. So which one's more efficient? You would use autoclaving. You use that moist heat over dry heat. But usually it's going to kill by an oxidation technique. So this can be your dry heating or incineration processes that are taking place. All right, we also have pasteurization, and pasteurization is not sterilization. This is actually just a way to remove and reduce spoilage and remove those unwanted pathogens. And usually what happens is you have high temperatures that are hitting the food or whatever it may be. So it's about 72 degrees Celsius for about 15 seconds. It's kind of like this almost flash hit, if you will. And this is how they would be able to remove some of those microorganisms, for instance, from milk. You can also see that they can use ultra high temperatures as well for like 140 degrees Celsius. And this is usually more common for organic milks or like, for instance, with coffee creamers that you can see. Now, you can also utilize low temperatures. And again, this is where we truly think about the idea of cytal versus static. In cytal, we're thinking about death. So using high temperatures would be very useful if you want to kill the organism, if you wanted something to be bactericidal. But if you wanted something to just inhibit growth, you would hear of static. So for instance, lower temperatures, microbes aren't able to grow in. So if you think about talking about those conditions, again, an organism has a max and a min. If you're using a minimal temperature and below that minimal temperature, it's going to inhibit their growth. So they won't be able to grow in those cold conditions, such as what you would see with refrigeration or even deep freezing, where they're just so suboptimal. They're not going to be able to have any growth take place. But if you put the organism in a hotter temperature, it would then continue on. It's not cytal, it's static. All right, we also have filtration processes as well. So you'll hear about HEPA filtration systems, and this is going to remove microorganisms that are less than 0.3 micrometers, which is about 3 nanometers. We also have liquid filtration as well, and it can get much smaller microorganisms. But again, when we think about the sizes of viruses are anywhere between 5 to 300 nanometers. And so the influenza virus is extremely small when we think about those sizes taking place. So these filtration processes are useful in some circumstances, but they're not necessarily useful in all circumstances. All right, now here's an idea to see for filtration. Again, if you were trying to filtrate a liquid, you have the sample in a flask, and then you can have this membrane filter, these HEPA filters. And there's a cotton plug to actually ensure sterility in the vacuum line. And then what is coming through this membrane filter has now been considered sterilized at this point. All right, we can also have radiation formats. You can have ionizing or non-ionizing radiation. Ionizing radiation is going to be what we consider like an X-ray or a gamma ray. This is really useful to use on certain organisms like meats and vegetables, and it's going to damage the DNA. It'll actually break the bonds of the DNA. This is also really useful for a lot of medical supplies that you'd see like syringes, and actually postal services actually use them for certain classes of mail as well. Versus non-ionizing radiation is going to utilize ultraviolet rays, and it does break the DNA, but it actually doesn't break the DNA bond. It creates what's known as a thymine dimer, and it blocks them from being able to replicate. So it doesn't penetrate. It's really only good for surface applications, but again, this can also be utilized as well. And then you can use microwaves as well, which is going to kill by heat, but this isn't necessarily for microbial growth. All right, so this just shows you that idea of ionizing radiation. So for instance, if you go to the grocery store at this moment in time, the reason that your strawberries actually look relatively fresh is because they have been treated. Versus if you can see with the ones that have not been treated with ionizing radiation, you have a bunch of moldy strawberries. The reason they look nice and fresh, even though they've been transported for long periods of time, is because they have been treated. All right, now we're going to think about chemical methods here really quickly. When we think about chemical methods, we think about these different actions that can take place of how they're going to be utilized on the microorganism. Some will change the permeability of the membrane. Remember that that cell membrane is really supposed to be there for protection. And if you damage that cell membrane, now you're going to have this leaky sort of component taking place, and eventually that cell membrane loses its integrity. So you can alter the cell membrane. You can also damage their proteins. So again, you can basically destroy their enzymes. And remember that their enzymes are very useful for all of their metabolism and their metabolic functions. So if you damage these enzymes, they are now rendered useless. Or you can actually damage their nucleic acids, such as their DNA. And this can be done with heat or radiation, and this can be lethal to the cells. All right, this is just a picture to show you an idea of the most resistant versus the least resistant. So the most resistant are going to be the hardier organisms and the ones that are very difficult to kill. As I said before, prions are really difficult to kill even with sterilization techniques. But when we do see sterilization techniques working, this would be useful for endospores of bacteria. Again, not all bacteria have endospores, but the ones that do, we want to be able to kill and remove. Your mycobacterium genera are really difficult to treat. Protozoal cysts, so those are those dormant phases. And then we get to kind of the more moderate, kind of easier to remove. And then we work our way down to the least resistant, which are those envelope viruses. All right, now again, thinking about this idea of microbial control and where some difficulty can come in. Gram-negative bacteria are more resistant than gram-positive bacteria, and it has to deal with that cell wall. Remember before that they have an outer membrane. Gram-positive bacteria have a very thick peptidoglycan cell wall, but they don't have that outer membrane. For Pseudomonas species, this actually helps them to become really easily resistant to biocides because they have that outer membrane, and they also have porins that are very selective to molecules. Again, we also have to worry about, for instance, Mycobacterium tuberculosis, the culprit of TB. It is made up of mycolic acid, and this is why we run that acid-fast stain instead of a gram stain. And that waxy component actually allows it to be very resistant against certain chemicals. All right, so kind of getting that idea of what's the most resistant and what's the most susceptible, we can run what's known as a Kirby-Bauer. And with a Kirby-Bauer, if you see this very high or large zone of inhibition, this shows you that this organism is susceptible to whatever this antimicrobial agent is. In this circumstance, it would be chlorine. So what happens here on these agar plates is you put a lot of bacteria. So this whole thing is covered in microorganism, just like this. And then we put these little discs all over the plate. So this one has chlorine, hexachlorophene, quat. And when we look at these guys here, we would incubate it for 24 to 48 hours. And if you see these holes, these are called zone of inhibitions. The greater the zone, the more susceptible a microorganism is to that agent. If you don't see a very big zone, for instance, like here, when we look at E. coli and hexachlorophene, or if we look at the Pseudomonas aeruginosa species, it's not going to be very effective. So those would not be antimicrobials that we would use. We would want something like, for instance, chlorine, or maybe chlorine with Pseudomonas aeruginosa. But again, Pseudomonas aeruginosa is very, very resistant. So maybe for this guy, a sterilization technique might even be needed. But these chemical methods, again, we can see how well these chemicals work utilizing this Kirby-Bauer plate. So the greater the zone, the greater the chance for susceptibility. All right, now talking about those prions, Prions are not cells. They're actually acellular. In fact, they're infectious proteins that are known to be heat and also what are known as protease resistant. So you can use to destroy them an autoclave in sodium hydroxide or straight up incineration. Those are the only techniques that are really useful against combating these guys. But prions are very, very dangerous. In fact, they're actually what are known to cause mad cow disease, Creutzfeldt-Jakob's disease, or Creutzfeldt-Jakob's disease. They also cause spongiform encephalopathy, or scrapie, as it's known as sheep. And prions are very tricky and very difficult to remove. All right, so thinking back on chemical disinfection, remember there is no perfect mechanism. There's no single disinfection technique that works for all circumstances. Maybe some will work for certain areas, but they don't work for all surfaces. Or maybe this one is appropriate for a toilet, but it's not appropriate that you would use on your countertop or on your floor or in your car. So there's no single disinfection technique that's appropriate for all. And the effect on microbes, again, can all be varying. So some will affect their cell walls, some attack the cell membrane or their proteins or their DNA. And these are really only useful for controlling microbial growth. They're not used for preventing disease. All right, so really quickly thinking about the idea of antimicrobial soap, is this a good thing or a bad thing? There's actually little evidence that shows this is actually good or aiding to human health. In fact, most of the products being used actually promote development of resistant microbes. That means that microbe does not have a large zone of inhibition. They have a very small zone, and they can combat that. They have the DNA or they have the genes that can code to still bypass some of these mechanisms. So the use of antimicrobial soaps should actually be really limited. It usually should be for food handlers, at-risk patients that are more immune compromised, such as someone that's had a transplant or someone that has a syndrome like AIDS, or again, caring for newborns. Those are really the only times that antimicrobial soaps should be used. Or again, we can even utilize them in the lab for safety purposes, but really they should be truly limited as much as possible because of this promotion of resistant microbes. And so the CDC just recommends hand washing with regular soap for 10 to 15 seconds. And again, the goal is to have least resistant microbes. But again, there's little evidence that is going to show that this actually helps human health. And again, thinking about this development of resistant microbes, one of the big culprits is trisglycan. So trisglycan is going to be actually disrupting plasma membranes, and it's an additive ingredient to a lot of antimicrobial soaps, toothpaste, deodorants. They're actually even incorporated into cutting boards and knife handles. And this widespread is actually showing reports of resistance. All right, so that's all I have for you for today. I hope you're having a wonderful day, and I will talk to you soon.